Sickle cell anemia is a disease that's caused by a mutation in one of the genes encoding for, or basically with the instructions for making hemoglobin, the protein that transports oxygen throughout our blood. More specifically, sickle cell anemia involves a mutation in the beta chain. So hemoglobin has 4 subunits, 2 alpha and 2 beta, and they kind of work as a team in this tetramer where each of those is holding on to a heme, which is basically this kind of like cofactor, this helper molecule that's bound to iron that binds to oxygen, and this is going to allow hemoglobin to pick up oxygen in your lungs and drop it off in your toes and places like that. So hemoglobin is really awesome, but what happens is that in sickle cell anemia, there's a mutation in one of those subunits. It doesn't prevent it from binding the oxygen and stuff. Instead, what it does is it affects the surface of the protein. It changes an amino acid from a glutamate, which is hydrophilic. It's one of these negatively charged, water-loved amino acid that's totally happy being on the surface. Water's happy with it being on the surface. This mutation, though, in sickle cell anemia changes that to a valine, which is one of our hydrophobic amino acids. It's water avoided. Water does not want to see it on the surface. And what happens in this mutation, in the deoxygenated state of hemoglobin, so when it doesn't have oxygen bound, the hemoglobin takes on this shape that's going to expose that valine. And it also exposes like a hydrophobic patch on the surface of hemoglobin. Now, if a valine, this hydrophobic valine on one hemoglobin, now it sees this hydrophobic patch on another hemoglobin, what's going to happen is that the water is going to kind of force those together, because this makes it so that the water doesn't have to like get tied up hanging out with too many of those. So it pushes those together, minimizing the water that's tied up around them. So it minimizes this exposed surface, and therefore, though, it kind of causes these hemoglobin molecules to link together. And because you have those 4 subunits, you're going to have those like 2 hydrophobic patches and 2 valines per hemoglobin. And so what's going to happen is that they're going to link up even more and more. You're going to get this polymerization. This polymerization is then going to cause the blood cells to kind of get misshapen, which is why you get that sickle shape. And those misshapen blood cells are then going to clog up blood vessels. And because you kind of have this cycling between these oxygenated and these deoxygenated states, and that deoxygenated state's the big problem, well, what's going to happen is that you're going to get polymerization and depolymerization and polymerization and depolymerization, which is going to totally stress the cells out. And so this is going to cause further problems. So you get those occluded blood vessels causing these painful crises, and you can get the like anemia and stuff because your blood cells are constantly undergoing this major stress. So today, let's look at the biochemistry of what's going on, look at the actual structures and things like this, as well as some of the strategies that are being employed to try to help counteract its effects. Let's just start by reviewing hemoglobin, and remember to look back at those past videos if you need a refresher. But basically, you have this protein that is a heterotetramer, so it has 4 subunits, and these subunits are not all the same, so we call it hetero. It has 2 alpha subunits and 2 beta subunits, at least in the adult hemoglobin. In the fetal hemoglobin, those beta subunits are going to be gamma subunits, and we'll talk a little bit more about this later and how this is a strategy to counteract sickle cell anemia. So each of those chains, so whether it's an alpha chain, whether it's a beta chain, both of them can hold on to a heme group, and that heme group is going to hold on to iron, and that iron is going to be what binds to the oxygen. What happens is that you can get disorders if you have mutations in the genes that are encoding for, or that basically have the instructions for making those alpha chains or making those beta chains. There are a number of different mutations, and some of them are more extreme than others. Some of them lead to mutations to like structural changes in the protein that we call structural hemoglobinopathies, such as is the case with sickle cell anemia, as we'll look at. And they can also be mutations that lead to reduced production of the beta globin, which would be like a beta globin thalassemia. So there are different types of disorders that involve hemoglobin, but we're going to be focusing in this video on sickle cell anemia, which is kind of like the classic example. In this
you have basically this beta S, and you end up with this HbS, which is like the kind of notation for that full hemoglobin in containing one of those beta S subunits. So in normal hemoglobin, what's going to happen is that there's this position on the beta chain that has a glutamate. So remember, glutamate is one of our acidic amino acids. It's got this negative charge. It's really hydrophilic. It likes to hang out with the water. The water likes to hang out with it. What happens is that a mutation in the mutation that's associated with sickle cell anemia swaps out that glutamate for a valine. And valine, that's one of our hydrophobic amino acids. So it's going to be avoided by the water. And so this amino acid would normally not be on the surface of a protein. But when we have this mutation, well, now we get this valine on the surface of a protein, and that's a big no-no. So this is going to cause the hemoglobin to polymerize and cause the blood cells to clump up and block blood vessels. So let's look in a little more detail at what's actually going on at the biochemical level and actually dig into those structures. So first, let's just review our hydrophobic effect. So remember that hydrophobic things are not afraid of water. Water just wants nothing to do with them. Water wants to form as many contacts with itself or with other polar things or charged things as it can. So it's totally happy to hang out with that glutamate. These are going to be very hydrophilic. It has a charge. Water's happy with that because water's got its polarity. But when you have something like valine, something that's nonpolar, well, here there's nothing to offer the water. And so water's like, I'm going to find other options. I'm going to maximize my interactions with water molecules and minimize my interactions with you. This leads the water to kind of clinch around those hydrophobic things. And so basically, if it can take that valine and it can squish that valine with something else hydrophobic, well, now instead of having to cover the valine and cover that other hydrophobic thing, if you push those together, well, now you're taking up less of the water, you're freeing more water, you're getting that release of entropy, hence the hydrophobic effect, leading to this favorable binding of nonpolar things to one another, even though those nonpolar things don't have an intrinsic desire to hang out with one another. So when we have a valine on the surface of a protein, basically we have to think about water having to kind of get tied up surrounding that valine. Because the water can't make favorable contacts with it, it's going to make favorable contacts with as many waters as possible. But this is kind of going to make it so that that water is stuck in place around the valine rather than being able to move around freely. If you can squish that valine to something else hydrophobic, well, then we have a better chance. We have more entropy, we have happier water. So if this valine is exposed, and if there's another hydrophobic thing around, well, then those valine and that hydrophobic thing combined, and this is going to release the water that was surrounding those hydrophobic things, leading to that increase in entropy. It turns out that you find this valine exposed more, and you find another hydrophobic patch exposed more in the T state of the hemoglobin, in the deoxygenated state. So hemoglobin has those two states. It's got the taut state, that is the low affinity, oxygen unbound form, and it's got the relaxed state. So our red, oxygen, red, relaxed. This is going to be the form in which oxygen is bound, and there's a high affinity for oxygen. This leads to that really cool cooperative binding effect, where basically when one oxygen binds, you stabilize in that R state, that high affinity form, making the other subunits bind to oxygen more quickly. But then if you release one oxygen, you switch to that T state, and now you can release the oxygen, dump it all out. And this allows us to take up oxygen in our lungs where we've got a lot of oxygen. We're going to be in that R state, and then transition to in our tissues where there's less oxygen. We're going to dump oxygen out. We dump one, we get into that top form, that T form, and we have a lower affinity, we dump it all out. And so this positive cooperativity makes it so that we can get to oxygen in our lungs, dump it out in our tissues, and do this over and over again. So remember, this is involving those structural changes. where we have like different interactions that are holding together the relaxed and the taut states. In that taut state, it's taut because it's got those salt bridges, those ionic interactions that are kind of helping stabilize that T form. And it turns out that in the T form, it actually is going to expose that valine better and expose that hydrophobic patch better. And so we'll look at this in a second. But basically what's going to happen then is when those are exposed in the T state, you're going to get them clumping together. And this is going to lead to these fibers forming that twist together in these long helical structures that are going to cause those red blood cells to get misshapen. So that's why you see those like sickle cells. So they actually look like sickles. That's where the name comes from. And this is caused by these forming. What's going to happen is basically because this is favored in the T state, but in the R state, that valine and the hydrophobic patch, they're more hidden better, then they're going to depolymerize when you're in that R state, when you have high oxygen. So you're going to get this polymerization and depolymerization and polymerization and depolymerization, which really stresses those cells out. Okay, so now let's look a little at the structure and look at where these mutations are occurring and how this is leading to the polymerization. I have a link I will share with you guys so that you can see these already aligned. But basically what I did was I took the structures for hemoglobin in the deoxygenated state and the oxygenated state and overlaid them in IC in 3D. If you want to do this yourself, basically just go to open IC in 3D, go to File, Align, Protein Complexes, 2 PDB structures. It'll ask you for the names. You put 2-HHB, which is the deoxygenated state, the T state, and then the 1-HHO, which is the oxygenated state. And so these are old structures, you can tell, because they have PDB accession codes that actually make sense. So we have like our hemoglobin, and then we have like an O where it's oxygenated. And basically these days, they just give them random numbers though. But when you do these, you'll get the structures overlined. Yours won't be in pretty colors, but this you can then adjust the colors. Remember to go to analysis, sequence, and annotations in order to be able to see all the details and to be able to see these. So you can see that for each of these, we basically have an alpha chain, a beta chain. This is going to be kind of like they have a like this one is labeled C, which is just the other alpha 1. And this one is labeled D, which is just the other beta. Or this one is labeled C, which is the other alpha. And this is labeled D, which is the other beta. But you have one of those for each of these 2 structures. And right now they're overlaid. But if you press A, you can alternate between them. I also colored them in the subunits in different colors. And so if you want the version with the colors, just go directly to that link rather than trying to align them yourself. But you can press A to alternate between the different structures. So here I'm showing the HB, which is the taut state. This is the state without the oxygen. Yeah, so when I aligned it, it like took away the heme groups, which was annoying. But basically, yeah. If you look here, this is that taut state, the deoxygenated state. And if you press A, now you get the oxygenated state. If you want to view them both, you can also go to view full structure. But I'm going to alternate between them to show you what's going on. So over here, what I have labeled this like GLU6. So basically, remember, this is the one that's going to be mutated. This is our E6. So this is going to become a valine in the mutated hemoglobin. But right now, what we have is basically we can see that it's going to be on the surface. And so when it's a valine, that's going to cause a problem. And it's mostly going to cause a problem, 1, if it's sticking out more, and 2, if there's something hydrophobic nearby that it can cluster with. So when I talk about nearby, though, it's going to be in a different hemoglobin copy. And this is going to lead to the polymerization, as we'll see. But where will we find that's nonpolar patch? Well, in the oxygenated state, right now, the residues that are going to be involved with that hydrophobic patch are actually kind of more facing the inside of the protein. They're more hidden. So a couple of the important ones that I've highlighted here, basically we have these 2. We have this leucine 88 And then we also have this phenylalanine 85. And so I have labeled them over here. So we've got this phenylalanine and we've got this leucine. Okay, so right now we're looking at the oxygenated state, and we can see that they're kind of facing up, they're kind of towards the inside of the protein. But what's going to happen if we go to the deoxygenated state? Well, now they're kind of sticking out more. They're going to be exposed more, and so they're going to be able to thereby provide a binding site for a valine that's going to be sticking out more now on another subunit. So they're hidden more in the oxygenated state, but then all of this is going to get exposed more in the deoxygenated state, making it easier for the polymerization to happen. There's this really cool animated kind of like tutorial you can go to through Molviviz. And basically it'll has a little information, and it's also going to have some views that are going to show you different things. So right now we're looking at the deoxygenated hemoglobin. And so this is our heme right here, and we're looking at our alpha and beta chains. What's going to happen is if you click on view 2, what you can see is it's going to show you where that valine is. So remember, that's going to be our V6 mutation, our EV6 mutation. And now what we can do is we can see that a small this hydrophobic patch is going to be highlighted. So in the yellow, they're showing us this hydrophobic patch, and this blue is the hemoglobin, and then this white is going to be the valine mutation. So the valine isn't going to bind to the nearby hydrophobic patch in the same hemoglobin subunit, but it can bind to another one in a different subunit. And so this is what's going to happen. The valine that is sticking out of one of the mutated hemoglobins is going to find a partner in the hydrophobic patch of a neighboring hemoglobin molecule. So this is just, before we were looking at the surface, and now it's going to show us in a little more detail. So we can see where we have these interactions occurring. We have that leucine, we have that phenylalanine. And remember, you have those on each of the beta subunits, which is why there's like two per tetramer. And so they're just going to show you a couple different views about the interactions. And you can see that you can kind of have these interactions occur, not just between those two hemoglobins, because you have those two beta chains per tetramer. What's going to happen is if a valine of one is being used with the hydrophobic patch of another, well, those are still going to have multiple other either hydrophobic patch, they're going to have another hydrophobic patch, or they're going to have another valine, and then they'll have two of whichever ones they weren't using up right now. Bottom line, you can get a lot of this polymerization happening, these hemoglobins linking up. And remember that because this is going to be, those are more exposed in that deoxygenated taut state, this is going to kind of polymerize and unpolymerize and polymerize and unpolymerize, causing a lot of problems. And I'll post a link to resources if you want to learn more, but hopefully this helps show you kind of like the biochemical basis. So the sickle cell mutation can be a really big problem, but it's typically just a problem if you have two copies of this beta S gene. So if you have one of the normal versions and one of the mutated versions, then basically you're heterozygous and you typically don't have symptoms. There are occasions where there's some symptoms, but there's also benefits. And so one of the benefits is that you're more resistant to like malaria. And so this has been evolutionarily conserved, this mutation, but if you have two bad copies of it, that's when you get the sickle cell anemia and can have really bad problems. There are also remember how we talked about there are different types of hemoglobinopathies. And so basically what can happen is that some people have like one sickle cell anemia gene, but then another of like a different hemoglobin mutation. And so there could be like mixed mutations and various forms of sickle cell diseases. But if you have a problem with your beta subunit, you have two chances to make it right. So you have you inherit two copies. If you have two bad copies, then you have a problem. One bad copy, typically you're okay. But if you have those two bad copies, there might be a third way to make it okay. So basically, how I mentioned, there's that hemoglobin. The fetal hemoglobin version differs in that it has gamma subunits instead of beta subunits. These gamma subunits are basically, they have a higher affinity for oxygen. And so the fetus makes them so that they can better grab up the oxygen after the mother has already kind of like used a bunch of it up. They need a high affinity for oxygen in order to take what's that little that's there. And so this is going to be this fetal form of hemoglobin that's going to stop being made, though, like after soon after birth and things like this. But it's not like your cells lose the instructions for making it. They just turn the making of it off. So your cells are always going to have the genetic instructions for making that gamma subunit. They're just not going to make it because of various regulatory mechanisms. So there are various strategies for treating sickle cell anemia, both genetically and pharmacologically, that act to try to counteract those breaks. So for example, there's this gene called BCL11A, which is basically going to prevent the expression of those gamma subunits. And there are genetic engineering strategies like using CRISPR to cut the gene for that, to cut and mutate the gene for that BCL11A to turn off those breaks and allow fetal hemoglobin to be made. And there are other genetic strategies. So there's a lot of things in the works to try to treat sickle cell anemia. Unfortunately, a lot of them are probably going to be very expensive and not accessible. But hopefully some of these findings can kind of lead to a more accessible treatment that can actually really help people both here and around the world. Changing from a glutamate to a valine doesn't just change the hydrophobicity, it also changes the charge of the hemoglobin. So remember that glutamate is negatively charged and valine is neutral. So for each of those swaps, you get a charge difference of 1, where you have one greater charge in the mutated version. And then remember, you have 2 of those beta chains per hemoglobin. So the sickle cell version of hemoglobin is going to have a charge of 2 more than the normal adult hemoglobin. As a result, if you have a way to separate proteins by charge, well, now you can actually separate the hemoglobin, these 2 different types of hemoglobins, and you can show that they're different. And this was really, really crucial to the findings of Linus Pauling et al. Basically, they were able to show that these versions of hemoglobin were different, showing that basically there was this genetic link between the inherited nature of this hemoglobinopathy, of the sickle cell anemia, and an actual physical change in the protein. So it was helping show that like differences in the genes actually led to differences in proteins. Then Ingram wanted to figure out, okay, well, what is the actual difference? Like, is there just a single difference? Is it always the same difference? Remember, these are like the early days of molecular biology, the early days of biochemistry, the early days of being able to connect structure and function and genes and proteins and all of this stuff. So basically, we had talked about separating the proteins at the big scale, like the whole protein. But you can also think about cutting up the protein and separating its pieces. If you do this, most of the pieces are going to be the same, because remember, we know now that there's only that one amino acid change, but they didn't know that then. And so basically they had to figure that out. And so Ingram was going to figure that out. So we cut up the protein, and then he wanted to find the difference between those pieces. So basically, he used electrophoresis to separate pieces by charge, but instead of using that weird, like, Thales apparatus that Pauling used, he's using papers, like wet paper. And he's using those chopped up protein pieces. You stick them on the paper, and then you apply opposite charges on either end of the paper. And the negatively charged pieces are going to go towards the positive end, and the positively charged pieces are going to go towards the negative ends. And so you're going to separate the pieces. But the ones that are like similar in charge are all going to be kind of clumped together. And basically, then you want to separate things further. So now you could use chromatography to basically to further separate them based on how they dissolve in the solvent. And so you can like separate in another dimension to kind of like spread them all out. The normal hemoglobin has a peptide with a more negative charge, so it'll travel further towards the positive charge. And then when you separate things out further, you should be able to identify the difference. You can look and see you can visualize the proteins using this anhydride-based method. You get this purpley color, so you can see the different spots on the paper. When he did this, he found a difference in one of the spots, showing that there was a difference in the protein. And so this wasn't enough, though, because he hadn't been able to analyze all of the protein. So we had shown that basically in that one portion that he could analyze, he only found one difference. But was there a difference in that part that was resistant to that initial cutter? And so remember, he had to cut up the protein into pieces in order to see if the differences between the pieces. Well, then what he had to do was he had to go and he had to take a different protease, so a different protein cutter, to try to cut up the pieces that he hadn't been able to cut up before. And when he did those, he saw that they were the same. And thus he was able to show that basically there's this one difference, this one amino acid change between the normal and the sickle version of hemoglobin. And I have a whole post on that if anyone is interested.